Hello, and welcome to today's Lunch and Learn. Today we're going to have a discussion around some special developability parameters for biologics. I'm Dr. Stephanie Call, and as part of my job on the product squad, I speak to a lot of customers working in the biologics field. Over the years, I've learned a lot of your most common concerns in the lab. For those of you already working in the early development space, you probably already know this. You're essentially asked to be a prophet. It's your job to select candidates from screens of up to thousands and to find the perfect buffer to keep it stable long-term for further research, development, and ultimately clinical use. I spend a lot of time working to make tools for helping you do this kind of predictive work and discussing with researchers how to improve what's already out there so they can do this work more efficiently. Today, I thought it would be helpful to discuss specifically how light scattering instrumentation is used to assess the self-association propensity of your biologic molecules. Self-association is the tendency for a molecule to stick to itself. It's a very important assessment criterion for those concerned about how their biologic candidates will behave long-term. In this early stage, you're often using low volume, low concentration biologics preps for assessment. However, once those treatments reach the clinic, they're administered at a much higher concentration, which alters physical properties such as viscosity and aggregation propensity. However, getting enough material to assess quality attributes at those high concentrations, it's often prohibitive at early development when there are lots of candidates to screen. So we'll talk more about what exactly self-association is, some different ways to assess it, and then show an example of data output. Then I'm going to share an interesting data set from Merck KGaA. They found that typical self-association parameters weren't always accurate enough to predict their self-association behavior, so they developed yet another method of predicting it. Overall, we'll be reviewing 3 great methods for predicting this self-association behavior. So first, I want to touch on what self-association is for those who may be new to or unfamiliar with the biologic developability space. Self-association is the tendency for like molecules to attract each other in solution. If the molecules are likely to self-associate, that means that they want to stick together. And if they're unlikely to self-associate, that is, they favor solvation, then they are driven apart by electrostatic forces that result from their chemical composition and buffer environment. Self-association is specifically used to predict how a biologic will behave when it is highly concentrated. As mentioned, early on, you're conducting extensive developability screens at these low sample concentrations. Now, the exact threshold for low varies from group to group, though generally I hear from people working with sample preps from about half a mg to maybe 2 mgs per mil. And for those of you who struggle to get lots of sample or who work with highly active or sensitive therapies, even these concentrations may seem high. However, once these therapeutics are ready for use in human patients, they have to be at incredibly high concentrations, upwards of 200 mg per mil. And that's because the dosage required in humans can be up to a whole gram in total. I really want to emphasize the importance of high concentration therapeutics for the clinic, since it's not always something top of mind when you're working in the early developability stages. So let's look at a real example. The recommended dose of the monoclonal antibody Rituxan for rheumatoid arthritis is 100 milligrams administered as 2 500 milligram doses. So if that Rituxan is kept at 1 mg per mil, which is a fairly typical working concentration for labs in early stages of biologic development, you're going to need 500 milliliter doses, which you might imagine doesn't actually work, as that much volume is going to disrupt your blood chemistry. Your body has about 5 liters of blood, so that's already almost 10% of the total volume. Now, if on the other hand, you have this Rituxan prepared at 200 mg per mil, which is a much more typical expectation for therapeutic administration, then you only need a 2.5 milliliter aliquot of it in order to administer. That is a lot easier for intravenous injection, and it means the patient won't be sitting there forever waiting for treatment to wrap up. Of course, drug administration requirements vary based on drug, and there are a lot more considerations for safety once you're actually in the clinic. But just at this example, you can see how it's better for patient health to have these lower volumes for injection. However, these high concentrations change the biophysical behaviors of the biologics in question, and it is very important to assess whether those changes will have negative impacts on stability. So let's talk a little bit more about the problems that can occur at high concentrations of protein-based therapeutics. First, if your molecules are more likely to stick together, they're also more likely to clump up and aggregate. Aggregation causes potentially devastating consequences to the protein-based therapeutics you're developing. Aggregated proteins are no longer active, which means overall your drug prep will be less effective. Further, aggregation is potentially toxic in patients. Finding byproducts or contaminants in a prep in the early stages of developability make it a bad candidate for scale-up and manufacture, as it is more likely to fail in the clinic due to those toxic effects. Another potential downfall of self-association is an increase in viscosity. When molecules are attracted to each other in a prep, they make the aqueous solution more viscous. Imagine working with an Eppendorf full of honey. Not only is it going to be difficult to pipette and work within the lab, but it's going to be difficult to move through any tubing or needles required for patient administration. Avoiding mixtures that will be high viscosity when they reach clinical concentrations is critical for the success of your complex therapeutics like biologics. All right, so now that I have described the pitfalls that occur if your biologic behaves poorly at high concentrations, let's talk about how to actually predict that behavior so you can optimize your formulations and avoid it. This is where light scattering comes in. Before we go on, I'd like to encourage you to check out our DLS Lunch and Learn titled How Biologics Teams Use Dynamic Light Scattering to Identify Leading Candidates. That's where I'm going to review some more basic experiments in DLS. Those type of experiments are often used during the first round of candidate triage to eliminate poor candidates based on obvious red flags in the preparation's purity. They're very fast to set up, and they can all be measured at single low concentrations, which means they're a great tool for eliminating the worst candidates from your libraries or the worst buffers from your screens. I often see researchers doing that kind of triage first in order to narrow down to a dozen or even fewer candidates for further assessment. Now, self-association experiments are the perfect next step for this initial triage. I will say you certainly can set these up as part of a large screen, but they require a bit more material compared to purity and colloidal stability screens. So it's important to be mindful of how much material you have available early on for all the constructs you're investigating. Light scattering techniques encompass both DLS and SLS measurements, that is, both dynamic and static light scattering. DLS uses the movement of molecules to find out information about their size and whether there are differently sized populations in the sample. SLS uses the average total amount of light deflected by the sample to inform on the average molecular weight of the sample, as well as its colloidal stability. Large particles scatter more light for SLS and move more slowly for DLS. Each light scattering technique is used to calculate a different parameter for self-association prediction. DLS gives the KD of the interaction. SLS gives the second virial coefficient, or B22. KD is a measurement of the dissociation constant, that is, the strength of interaction between two molecules, in this case, two identical biological molecules in solution. A lower KD value indicates stronger attraction between molecules and is therefore less desirable. B22 derives from concentration dependence of osmotic pressure, which informs on the strength and the nature of protein-protein interactions more globally. A negative B22 indicates attractive interactions and should be avoided. I don't want to get too into the mathematics of how either of these are calculated. We have some deeper information in a DLS ebook, or you could reach out to discuss with one of our technical specialists later. But to keep it very simple, for both KD and B22 experiments, you will prepare replicates of your sample at about 5 different concentrations, from half a mg per mil to maybe 10 or 20 mg per mil. A light scattering measurement is conducted on each individual sample, and depending on which type of light scattering you're talking about, a graph is produced that is concentration versus a dependent variable that correlates with each technique. Whether the slope of the resulting fit line is positive or negative, and how positive or negative it is, indicates the tendency towards self-association. Let's look at a real-life example. We're just going to stick to KD for now. Here we have the same monoclonal antibody in two different buffers. The only difference is whether or not there is salt in the buffer. Salt often helps with the solubility of a protein when the protein is at lower concentrations, as it interrupts the electrostatic interactions between two protein molecules. However, as the protein gets higher in concentration, that same electrostatic protection may actually start to become a detriment as more protein molecules start competing for water molecules in order to remain solvated. The interplay between salt concentration, pH, additives, and the protein itself all have dramatic impacts on the protein solvation, but they really do vary from protein to protein. This is another reason why it is so important to assess self-association prior to scale-up. All right, so here the sample without salt has a positive slope and therefore positive KD, while the salt-containing sample has a negative slope or negative KD. Therefore, if you were choosing between these two formulation buffers, it's most ideal to select the former one without the high salt concentration. A few notes on sample prep here. For SLS in particular, at these low concentrations, there can be a lot of measured variability from the scattered light, which makes it difficult to confidently fit a line. It's a good idea to filter your samples first to ensure that you are only measuring the effects of your protein sample and its buffer. Remember, you've likely already eliminated the worst, most impure samples at an earlier triage stage. And don't forget to prepare the filter by first pushing some buffer through it before collecting your sample. Further, we always recommend at least duplicates, if not triplicates, at the different concentrations. Mix your samples thoroughly but gently via pipette. Never ever vortex proteins. And as you dilute out, be very cautious that you're not picking up lots of extra sample on the outside of your pipette tip and transferring it to the next tube. Okay, now with all of that, let's actually talk about an alternative method for predicting self-association. This methodology was developed by Dr. Melanie Hoffman and her colleagues at Merck KGaA, and the methodology will be referred to as SLS deviation from linearity. I highly encourage you to read the paper. The publication is linked below. They also worked with us to develop an app note, and I will be sharing some of that data here. The group required a different approach to self-association prediction. They found that KD and B22 measurements weren't always identifying the optimal buffers for their different constructs. As with any prediction model, there's never a perfect answer. And this also applies to how well these low concentration methodologies work for predicting high concentration behavior. Dr. Hoffman's group came up with this alternative method, which still uses light scattering, but it improved their predictive success by including a higher concentration method measurement as well. Dr. Hoffman's group preferred method starts similarly to the KD or B22 technique by preparing the sample at different concentrations. They selected 2.5 mg per ml up to 15 mg per ml, and then they measured it with a light scattering instrument. Here they used a plot of concentration versus scattering intensity, which is an SLS parameter. Then a best fit line is fit through the data and extrapolated out past that 15 mg per ml concentration. Finally, they prepare one last sample at a higher concentration of 40 mg per ml. They found that this is an ideal concentration for this type of experiment, as it's past the point where the linearity of those low concentration samples break down, but it's not so high that it's difficult and resource intensive to make a small aliquot at this concentration for measurement. So the scattering intensity at 40 mg per ml is measured with SLS, plotted on this graph with the extrapolated line. If that point is below the line, that indicates less propensity for self-association, which is good. And if it is above the line, that is a sign of increased tendency to self-associate, which is bad. The further from the fit line, the more extreme the effect. They use this information to rank several candidates. Let's take a look at a few. Let's start by actually looking at how this SLS deviation from linearity method stacks up against a more typical KD measurement. Here we have a monoclonal antibody in several different buffers. The KDs were all found to be negative, which generally indicates self-association and would generally tell them that more buffer screening is required. However, this group had already done additional long-term stability studies, and they found that the pH 5.5 buffers did an acceptable job of keeping the monoclonal antibodies stable for long-term. So let's take a closer look at the KD values for the antibody at these pH 5.5 buffers. Both of them give negative KD values, but the group knew these buffers actually stabilize the protein in storage. So now they turn to their SLS deviation from linearity measurements. Here we see that in both instances, there is a negative deviation from the linearity, which is good. Based on that selection criteria, the group would be likely to select these buffers rather than reformulate as those KD experiments may have suggested. The group therefore concluded that for their needs, the SLS deviation from linearity measurement was a better predictor of high concentration behavior. I'd like to quickly point something out about light scattering. As I suggested during the sample prep section, scattering data measurements are quite prone to variability. Aggregate clumps or dirt potentially contaminate and interrupt accurate readings. Lower concentration measurements are simply noisier. That's why it's important to select an instrument with good resolution and high data quality so that you're ensuring good reproducibility. Here we have Nanotemper's Prometheus Panta compared to a DLS plate reader. Each dilution series was prepared twice, then measured in triplicate on the respective instruments. Note that there is far less deviation for the Panta measurement. This makes it easier to confidently predict the SLS deviation from linearity with these high-quality, reproducible measurements. All right, so last I just want to quickly show that this approach isn't limited to full-length monoclonal antibodies. Here we have a brief example of the same experiment applied to an FC fusion construct screened with 4 different buffers. The same histidine pH 5.5 buffers give negative deviation from linearity compared to the higher pH buffers. This just goes to show that the predictive measurements apply to all types of biologics constructs and not just monoclonal antibodies. In conclusion, self-association measurements are critical at early stages, as they have outsized impact on the long-term developability of a therapy destined for patient use. Though self-association experiments are a bit more time and sample consuming to set up, they're well worth it in terms of determining if your biologic will not be aggregated or viscous upon clinical administration. So thanks for joining me today to learn about self-association measurements and how they help you understand how your biologic will behave once it leaves your hands and reaches the clinic. Thanks.